Welcome to the University and Institute of Advanced Research, Gandhinagar, Gujarat. I, Ashiva Bhardwaj, represent the Laboratory of Molecular Biology of Disease, a part of Department of Human Health and Disease in School of Biological Sciences and Biotechnology. The laboratory became functional in 2007 under the mentorship of the founder director of Indian Institute of Advanced Research, Dr. Amit Ghosh. The laboratory is focused on research pertaining to molecular epidemiology of antibiotic resistance in two pathogens at this moment, Vibrio species and Shigella species. India, as you all know, is endemic in cholera, and diarrheal diseases have been a major burden for social and economic growth of our country. The problem of disease management gets aggravated with the appearance of multi-drug resistance, commonly known as MDR. As you may be aware, the phenomena of MDR has created a bottleneck in the management of many common diseases that ail humankind since long. These may be tuberculosis, cancer, cholera, typhoid, or any disease which is a problem these days.
Welcome to University and Institute of Advanced Research, Gandhinagar, Gujarat. I am Dr. Dheeraj Naik, presently working as an environmental scientist, plant ecophysiologist, heading the Department of Environmental Science. Presently, our group is working in understanding the integrative response of CO2 and water budgeting of desert and semi-arid grassland ecosystem. This is micrometeorological technique commonly known as eddy covariance. It measures continuous fluxes of CO2, carbon dioxide, water and energy and many other atmospherical parameters which help us to understand whether the desert or semi-arid grassland ecosystem whether it is acting as a source or sink. This type of continuous information will help us to give us the annual budget of carbon and water.
I am Dr. Rajani Nadgowda, Professor Emeritus at this university. heading the project on isolation of secondary metabolites from certain important medicinal plants. My interest is to isolate the active compounds in a pure form from the cell culture lines of these plants. This is the culture. You see a different color. This culture, when grown in different conditions, is entirely a different callus, as you see. And here is one more callus line. So likewise, we have 5 or 6 different callus cultures, giving 6 compounds in this laboratory.
Myself, Dr. Anand Krishna Tiwari. I am doing research in the field of genetics and developmental biology. Our laboratory uses Drosophila melanogaster as a model organism of developmental biology. Drosophila melanogaster, a tiny insect of 3 mm, commonly known as fruit fly, is more than 100 years old model organism and perhaps the oldest model used for genetics and biological studies.
I am a junior research fellow at Bioinformatics and Structural Biology Group. The well-equipped Bioinformatics Center at University and Institute of Advanced Research has been established by the Department of Biotechnology, Ministry of Science and Technology, Government of India as Distributed Information Sub-Center to act as a nodal point for dissemination of bioinformatics information. Hands-on bioinformatics training programs are conducted regularly by the department, which is well attended by the scientific community in and around Gujarat.
I'm a PhD candidate working on cancer biology at Department of Cell Biology at University and Institute of Advanced Research, Gandhinagar, under the support of the Puri Foundation for Education in India. I presented a public lecture on aspects of treatment of cancer at the annual meeting of European Molecular Biology Organization at Amsterdam in 2013.
afternoon, everybody. I am Khyati Mehta. I am a student of Communicative English at UIAR, Gandhinagar. I am thankful to Puri Foundation for providing me true education.